Hi everyone, we are in week 7 and this week we're going to talk about the physical factors that are required for microbial growth. So there are 3 important physical factors in the environment that can affect how well and how quickly microbes can grow under certain conditions. So the first physical factor is temperature. The second is osmotic pressure or salinity, which is the salt concentration in the environment. And the third factor is pH. So we're going to go through each of those factors. The first physical factor is temperature. And every microbe has a minimum, a maximum, and an optimum growth temperature. And that range is called the cardinal temperature range for that organism. So the minimum and the maximum temperatures define the growth and development. So, in other words, below the minimum temperature, that microbe wouldn't be able to grow. Above that maximum temperature, the microbe wouldn't be able to grow. So that's the range in which it's possible for the microbe to grow. But the optimum temperature, as you can see in the graph here, is the temperature that is the best conditions for that microbe. So it's the temperature at which the growth proceeds at the most rapid rate. And the reason for that is because the enzymes within microbes are sensitive to temperatures. And above or below certain temperatures, those enzymes won't work. So what happens to those bacteria outside of that range? Well, below the minimum temperature, bacteria are metabolically inactive. So that means they're not growing. And the term bacteriostatic means that it prevents the growth and reproduction of bacteria or slowing the growth of the bacteria. So for this you can think of food spoilage in a refrigerator. It's slowed by the low temperatures. Bacteria may still be alive and able to survive at the low temperatures, but the growth of the bacteria is very much slowed by the low temperatures. And then above the maximum temperatures, most enzymes get destroyed above a certain temperature. So that would cause the microbes to die. So it's not just that they're able to survive and not grow, but in this case, above maximum temperature, they would die. So that's called bactericidal. Bactericidal means it kills the bacteria. There are 3 groups of bacteria based on the temperature ranges that they survive and grow best at. So the first group are the psychrophiles, and the temperature that they grow at are in the range of -5 to 20 degrees Celsius. So below freezing up to around room temperature. So you might find those kind of bacteria in a refrigerator or freezer. And what do they usually cause? They usually cause food spoilage. The second group of bacteria based on temperature are the mesophiles, and they grow in the temperature range of 20 to 45 degrees Celsius. So what's important about that range? Well, the significance of that is that that's the range that's room temperature. 20 degrees Celsius is room temperature, and also including the range of body temperature, which is 37 degrees Celsius. So the bacteria that are human pathogens are mesophiles. They survive best at body temperature. And also the bacteria of the human microbiome are also mesophiles. They live inside your body at 37 degrees. And the third group of bacteria based on temperature are the thermophiles. And the thermophiles are the group that really prefer the very, very warm temperatures. So they grow above 45 degrees Celsius. So that's above 113 degrees Fahrenheit. So they are bacteria that you would find in hot springs and hydrothermal vents and really, really hot places. And within the thermophiles, there's 3 different classifications. The first is the facultative thermophile, and they thrive at those high temperatures, but they can, if they need to, they can also survive below 45 degrees Celsius. So they grow best above 45, but they can survive below 45. On the other hand, the obligate thermophile can only survive above 45 degrees Celsius. So they wouldn't even be alive below that temperature. So to remember that, for obligate, think of the word obligated. It means you have to, right? So for obligate thermophile, they have to be above 45 degrees Celsius. And then the third classification is the extreme thermophile. So they prefer the really hot temperatures above 65 degrees Celsius or 149 degrees Fahrenheit. And they survive in really very hot places like hydrothermal vents. So we talked about how every bacteria has its optimum growth temperature, but that doesn't mean that every single enzyme within that bacteria necessarily agrees with that optimal growth temperature. So the optimal growth temperature is not necessarily ideal for every single enzyme in that organism. So an example of that is something you can see in the bacteria Serratia marcescens. Its optimal growth temperature for the bacteria is 37 degrees. But there's an enzyme in the bacteria called prodigiosin, which produces a red pigment, and that enzyme is only active below 30 degrees Celsius. So if you see red colonies growing in that organism, it means that the Serratia was grown at room temperature at 20 degrees instead of at 37 degrees in an incubator. Now let's move on to the second physical factor, which is osmotic pressure or salinity, or the concentration of salt in the environment. So I want to start by reviewing the term osmosis. Osmosis is the movement of water through a selectively permeable membrane from an area of high water concentration to an area of low water concentration. And water moves across membranes like cell membranes in response to the solute concentration inside and outside of that cell. So the solute is whatever is dissolved in the water. So for example, salt or sugar or whatever it may be that's dissolved in the water. So what happens is, and you can see in this picture, on the one side that has the lower concentration of salt, that's considered a higher water concentration. And the other side that has a higher salt is considered a lower water concentration. So what happens is the water actually moves through that membrane, and that membrane represents a cell membrane, and it moves through the membrane and actually causes an increase in the volume on the other side because it moves in response to the concentration of the solute and actually dilutes the solute. So the water is moving from its own high water concentration to the lower water concentration until the concentration becomes equal on both sides of the membrane. So why do we care about how water flows into and out of a cell in response to the solute in the environment? Why does that matter? Well, it's actually really important to the state that the cell is in. So, for example, if too much water flows into a cell, it could cause it to burst. If too much water flows out of a cell, it can cause it to be shriveled. So the osmotic pressure in a cell is really important to its survival. So the movement of water through a cell membrane depends on the amount of solute in the cell's environment. So an isotonic environment means that the concentration of the solute is the same on both sides of the cell membrane. So that's represented by the middle picture here. It's an isotonic environment, and that means that the amount of water that flows into the cell and the amount of water that flows out of the cell are about balanced, and it keeps the cell in a normal state. So an isotonic environment is a healthy state for most cells. And it's really important in the medical field. Most IV fluids are isotonic solutions, meaning they have the same concentration of solutes as the blood plasma, so that the blood cells don't shrivel or lyse. The second type of environment is called the hypertonic environment, and that's represented here in the darker blue picture. Hyper meaning high, so that's the concentration of solute is higher than inside the cell. So what happens to water if the concentration of the solute is higher in the environment? The water is going to want to dilute that, and it's going to flow out of the cell. So that causes the cell to become shriveled. And then a hypotonic environment, hypo meaning low, is represented by the first picture here. The concentration of solute is lower than inside the cell. So what happens in that situation, as you can see in the first picture, water is going to flow into the cell to try to dilute that higher salt concentration that's inside the cell, and that can cause the cell to burst. So the concentration of salt in a cell's environment is really important. And most cells prefer an environment with 0.5 to 3% salinity. But if they're able to grow in a salt content that's higher than 3%, then the bacteria are called halophiles. So above 3% normally would be inhibitory for most bacteria, but there are certain bacteria that can thrive above that. So within the halophiles, there's 3 classifications. Again, there's the facultative halophile, and they thrive at a salinity above 3%, but can survive below 3%. So they grow best above 3%, but they can survive below 3%. The obligate halophile, again, think of obligated. They have to have a salinity above 3%. And then there's the extreme halophiles that thrive above 30% salinity. So the picture here showing some extreme thermophiles in salt ponds. And finally, let's move on to the third physical factor, which is pH. So most bacteria prefer a pH around 7, or around neutral. And they are called neutrophiles. So neutrophiles are bacteria that thrive in environments with a neutral pH. So that's most bacteria. So the range that they can withstand is between a pH of 5.5 to 8.5. But optimally, they grow around a pH of 7, which is neutral. And that's the most common type of bacteria, and you can really find them everywhere. Acidophiles are bacteria that thrive in highly acidic environments. So the type of pH that they would withstand, as you can see in the picture here, is a pH in the range of 1 to 5.5, so very acidic environments. And optimally, the optimal growth is around a pH of 3. And where might you find them? Well, your gastrointestinal tract is an acidic environment, and they are very common in the gastrointestinal tract. And then alkalophiles thrive in highly alkaline or basic environments with a high pH. So alkalophiles are less common. There aren't very many bacteria that are alkalophiles. And they withstand a pH in the range of 8.5 to 11.5, so a very high pH, very basic, and they grow optimally around a pH of 10. So as I said, they're not very common, and that is a good thing because that means that we can effectively use bleach. Bleach is a very strong alkaline, a very strong base with a pH of 11. So that means that bleach is a very effective way to kill most bacteria.